So we're going to review the third week, which is also the concluding part of the first inquiry case. Most of this week we spent on evolutionary trees or phylogenies, looking at what they mean, how to read them, how to use them to ask questions about trait evolution and things like that. And you'll recall the first thing we did was an activity on putting together a tree using our pipe cleaner. So that's just a way of having a little bit of hands-on experience with trees and rotating nodes and all the rest of that. So you'll remember that we built our tree to fully resolve topology just with a couple of pieces of information. So we had to understand what we mean by outgroup. Then we talked about sister taxa, and then we talked about which groups are more closely related to others within that. And with those three bits of information, we were able to get a fully resolved tree with no polytomies, meaning that all the branching was depicted. We played around a little bit with reformatting the way the tree is drawn, and that doesn't matter if the topology is not affected. And we talked about rotating internal nodes, which is one of the reasons that you can't read any information across the tips of the tree. So the order of species that they show up from left to right across the tree in a format like this is meaningless because you can rotate any of those internal nodes. We talked about what a clade is. A clade is a monophyletic group. It's a group that includes the ancestor and all of its descendants. It's not leaving out any of the descendants. If it's not monophyletic, if it leaves out some of the descendants, we would call that paraphyletic. So things like reptiles are paraphyletic because it leaves out birds. And we asked you to be able to identify what is a clade and which is not a clade. You could also look at how many things on here are clades, or how can you assess that. And just remember that a clade is the ancestor and all of its descendants. It doesn't matter at what scale on the tree, as long as it meets those criteria. We talked a little bit about speciation and how it's a splitting of lineages, and from our previous discussion you know that that means essentially something has to block gene flow, because if you have a lot of gene flow you're going to get the two populations becoming more similar or remaining similar, not diverging. And then there were a number of different misconceptions about reading trees, some of them having to do with reading across the tips.
So a lot of questions actually that we had after taking a look at this diagram. Some of you were confused as to how to calculate which ones are more closely related. And the very simple way of looking at it is that the more recently they shared a common ancestor, the more closely related they are. So in order to be able to answer the question, then what you need to do is take a look at the comparison groups and ask yourself where the most recent common ancestor is for those two groups, and whichever one is higher up or further along on the tree, then the more closely related they are.
Yeah.
So if you take a look at the human and the mouse, you can see that they share a common ancestor fairly recently, and certainly more recently than the mouse and the lizard. The lizard and the mouse have a common ancestor, so they are related. In fact, everything then is related. But the human and the mouse share a more recent common ancestor, so they are more closely related.
So be very careful with that, not to just count the number of nodes, because that depends a lot on which species we include in the tree. So I could put a lot of nodes on the fish side. You can't just count the number of nodes. It goes by how recent the ancestor is, and another way of saying that is, do they share an ancestor that isn't shared by others? So for example, if you have a brother or sister, your siblings, you have the same parents, your cousin does not have the same parents. The next closest shared ancestor with a cousin is your grandparents. So the more you have to go back in the generations, or the more branching points you have to go back to find a shared ancestor, the more distantly related you are. It's not just the sheer number of nodes on the tree, so just watch out for that.
So you're more closely related to your sibling than you are to your cousin.
That's right, because you share an ancestor with your sibling that you do not share with your cousin. Good?
Good.
Okay,
Yeah.
so we talked a little bit about time, the amount of time, and you have to bear in mind that initially these ancestors were one thing, one population, one species, and then there was a divergence. And then maybe there was more of those episodes of speciation along one branch or the other, or both. But the amount of time, talking about number of years, since those two lineages split is the same along both of those two lineages, if they both still exist. So the ancestor here, for example, that's, say, the ancestor of all bony vertebrates, the amount of time leading to a modern fish species and the amount of time leading to, say, humans is exactly the same, because they converge at that point in time where they had a common ancestor. There may be a lot more speciation, a lot more physical change, a lot more other things happening in one side or the other, but the amount of absolute time is exactly the same, and that's always true when you have when you go back to a common ancestor. And we cautioned you to be very careful about how you interpret things like the outgroup. Outgroup does not mean ancestral. It does not mean it looks like the ancestor. It doesn't mean it's more primitive than the other things. The outgroup just means it's the least closely related to the in-group. And there can be lots of evolutionary change that has happened in that lineage. It may just not be depicted in the tree. So this echinoderm tree with the sea star, sea cucumber, sea urchins, humans are a legitimate outgroup to that. You wouldn't therefore say that the ancestor of all of these groups looked like a human, right? So be very careful about that.
So just to apply it directly to the echinoderms, what we're trying to demonstrate is the fact that we cannot say with any certainty what any of those ancestors looked like. And we certainly wouldn't say that the ancestor on the very bottom, where the humans and the echinoderms come together, was either an echinoderm-like thing or a human-like thing.
No, it wasn't.
There isn't any information there to be able to tell us what it was.
And in fact, you could, with great confidence, say
Say
it
it's
was
not.
not an echinoderm or a vertebrate,
Yeah.
right? It was something that doesn't exist anymore. Sometimes you have fossil data, and you can say, okay, the earliest vertebrate looked like this or that, but remember that ancestor is gone, and we infer its existence from these relationships. Okay. We asked you to contemplate this question about are we descended from monkeys, and this has to do with the concept of paraphyletic groups and naming groups with in biology if you want to have them be evolutionarily meaningful. So what you need to do to answer this question is think about what groups does the word monkey refer to? Is that a clade? And if you wanted it to be a clade, what would you have to do with the term monkey? To whom would it have to apply to make it a proper clade? Then you can ask, are we descended from monkeys? Obviously not modern monkeys. Are we descended from monkeys? Is there any way you could answer that question? Yes, based on your understanding of phylogenies and paraphyly and stuff like that. So we'll let you think about that one. We also wanted you to think a little bit about phylogenetic relationships and how they're put together with different bits of information. Spend more time on that in the next bit, which we'll talk about. There's a few terms you need to review. They all have to do with the evolution of traits and groupings that arise by speciation. And Dr. Jacobs suggested that you try to organize them into sort of related concepts so that you can keep in your mind what you're talking about. So instead of just memorizing terms, try to organize the meaning of them in some way that makes sense to you. So then we did some application of concepts. So one of the questions that you'll see as a practice question for the exam on the discussion forum was looking at an observation. In this case, you find some mussels with a simple lure, some with a complex lure. As a biologist, what are some of your available hypotheses to account for that difference? And not just, abstract ones, but maybe this is the actual situation in that habitat. Maybe this is the actual historical thing that happened in that population. So you need to be able to phrase and evaluate those kinds of hypotheses. So that's what this is about. And again, this is on the discussion forum, so it would be a very good thing to work through that as you're studying for the exam. And then we worked a little bit on trait evolution as mapped onto a phylogeny, so using evolutionary trees to understand when traits arose, when they were lost, and those sorts of issues. We talked about an obvious case like this, where you have basically a tree of vertebrates, and you can say, well, they all are vertebrates, so by definition they have vertebrae. That probably evolved once in the earliest ancestor, and they've all just kept that, whereas something like feathers has only evolved in birds, or fur has only evolved in mammals. whereas four limbs has evolved in the ancestor of amphibians, quote, reptiles and birds, and mammals, and then is retained in those groups. There is loss, so four limbs are not found in whales, for example. Then we asked you to try to build a phylogeny based on some morphological characteristics. And we started you off with just shape and color of the shell, and we asked you to do your best to come up with some kind of hypothesis about how they're related based on these traits. And then we asked you what the implications were. So if you started with, say, shell shape, then you were automatically assuming that color must be able to change much more easily than shape can. And the reverse would be true if you started with color. So we want you to realize that when you decide to classify things based on one characteristic, you're automatically making assumptions about the others. Like it follows directly from that. We gave you some more information about their lures, and again asked you to start talking about how these traits would evolve. We told you about their hosts and asked you to look for patterns, and things that came up in class were the number of fish hosts versus lure complexity seems to be related. We talked about trade-offs between having a complex lure and a simple lure. You can attract more fish with more types of fish with a simple lure, but you're not as good at attracting any particular fish as you are with a complex one. Risks of extinction and all those kinds of issues that you should definitely continue to explore. And then we said, let's give you the phylogeny that is built with genetic data or something and ask you to interpret it. And this is a question that we went through in class, and we're just going to spend a second on it because about half the class got it right, I think, and half were sort of all across the rest of the board. So let's just go through. This is also how you can study for this and how you should write the exam, which is, again, look at each option on its own and evaluate it on its own, and then decide on that one, move to the next one. But be able to justify in your own mind why you chose or did not choose that particular answer. So let's look at it. So we've got this tree with these 6 species of mussels. You can see the blue, green, and the red are each sister taxa. The larger lures are part of this clade, and then the simpler lures are part of a separate clade over here. First statement, we'll just look at this one. So ancestor X could not have been blue. If ancestor X were blue, it would mean that twice there was a change from blue to green. Minimum amount of change, right? So if this was blue, they split, now there's 2 green ones, so there's been at least 2 changes from blue to green. Is that impossible? No, that's not impossible. That could certainly have happened.
And you can then take that scenario and you can kind of play with it a little bit and say, what color is ancestor X most likely to be?
Yes.
But it doesn't mean that any of the other interpretations are impossible, right?
Yeah.
So the ancestor X could be fuchsia.
Yeah.
We don't have enough information to know what it is. We do have enough information to say what it's likely to have been.
Then you can take that to the next level and say, what color is W likely to have been? It's not as obvious as answering what X was, and you should understand why that is, so as you review.
And then further, what color was Z?
Was Z,
Yeah.
which we'll get to, I
So the
think. Yes.
likelihood decreases as we get further and further away.
The ability to
Yeah.
determine, yeah. Look at B. Shell shape has changed at least 3 times. So that means that if we assume different characteristics for the ancestor, we should see evidence that there's been at least 3 changes in shell shape, no matter which one we assume. So the best way to do this kind of analysis is to take one of the 3 options. So you've got tall, medium, and short shells, and just choose whichever one you want and start with an assumption that the ancestor here, Z, had that. How many times does it have to have changed to give you what you see in the current species, the modern species on the tips? So let's just take one. Tall. If you imagine Z had a tall shell, then we could say, okay, it kept tall, it kept tall, it kept tall to there, but there must have been one change here. Kept it all the way to here, that's another change here is 2. And even if it kept tall all the way to here, that's 2 more. If it changed to medium on the way, then it would have been kept, but then it changed once more again. So that's 4 changes if we start with tall. If we start with medium, we can say medium was kept, but there has to be 1, 2, 3 changes, right? With 4, because these are neither medium. Or did I get confused? Anyway, if you start with the short one, same idea. Short was kept, but there's 1 change there. There has to be 2 changes there, and a change there is 4. So I think in the end, actually, the minimum number of changes is 4. I'd suggest you work through this carefully. And then, as we recommend on the discussion forum for studying, change some of them. Put this green one here. Do the exercise again. Now, how many times does it have to have changed? So go through the activity of not just evaluating these as it is here, but change it and see if you can interpret it differently. So B was the answer. It's no matter what starting point I have, it's in fact at least 4 changes in shell shape, and that's assuming that Z had one of the existing ones. It may have had something else. In fact, it may have been a square for all we know, right? So it's at least 4. So B is correct.
A kind of added question that you might want to now play with is seeing whether or not your tree can tell you about the maximum number of changes that have occurred in this lineage. It's kind of a trick question, but if you play with it and try to understand, then I think it'll capture pretty much everything that we covered in terms of tree thinking and tree reading and reading trees. So try to answer that one.
Yeah, or if you come up with a really high number for that, then what are you assuming about how that trait evolves? So anyway, that's a very good idea. Let's just look at C. So we're pretty sure B is correct, but we're not going to stop as soon as we find one. We're going to always evaluate each one on its own merits. So let's look at C. It's very likely, when you see very likely, you should say, boy, there better be quite compelling evidence for this if they say very likely, that Y lost a complex lure. It's very likely that Y lost a complex lure. You might choose that one if you're thinking, hey, the most species have complex lures, so that must be the one that changed. That's not how you read that tree. So again, start with the different scenarios. So a simple lure here would mean that it maintained simple there, and there was maybe a complex lure evolved here, and then some modifications. That's perfectly plausible. Or you could have a complex lure evolving here, so that W had initially simple, and then there was a split to more complex. All those things are possible scenarios. There's no really obvious reason to interpret this tree as saying that had lost a complex lure. It may have maintained an ancestrally simple lure.
But try to now draw a tree where that becomes a true statement.
Yes.
So the idea being that then that trait, that complex lure, would be nested, or sorry, the loss of the complex lure would be nested within a clade of those that had complex lures. And in that case, a recent ancestor would be more likely to have had it.
So what would this tree look like? Where would the red ones go on this tree to make that statement true? Because there is a way to do it. All you have to do is swap the red ones with 2 other ones and you'd see it. Ancestor W, this is D, very likely had a small lure. Now the reason people get caught with this one is they tend to think, oh, these guys are simple and they're the outgroup to this other one, so this is what the ancestor was like. Remember, you can't do that, right? Think humans and echinoderms. So we don't know whether this was a small lure here, and we certainly don't know whether it was here. This could have evolved a complex lure and then lost it. It could have evolved, it could have been simple here, it could have been complex here. We don't know. So there's nothing about this tree that tells us it's very likely that this was a simple lure, unless you're misreading the tree to assume that the ancestor must have been simple because these ones are simple here. And then E, ancestor Z was very likely red with a large lure. So this is a misconception where the ancestor is somehow intermediate between the modern species. So it has to have some traits from each. So if this is the ancestor, it must have had the color from the red and the lure from this side, and then that's how you got the divergence. That's not true at all. This could have been purple with no lure whatsoever, and then evolved those traits since then. Okay, so you can't take it as an average. The ancestor is not an average. The ancestor might have been something quite distinct. Again, the ancestor of humans and echinoderms was not half human, half echinoderm, right? It was something that was neither of those groups. So this is one of your study questions for the exam. some tips on how to use it effectively for studying, and you've got some hints of that just here as well about flipping things around and reevaluating and what would it have to look like for that to be correct and stuff. And we talked a little bit about trait loss and how you might identify examples of that. As Dr. Jacobs just said, often if you see a missing trait, a species that's nested within a bunch that have the trait, that's a good sign that that trait has been lost. It's not necessarily the case, but then you'd be assuming that there were multiple cases of it evolving in the first place. And we gave you an example from birds losing flight, humans losing fur. There's lots and lots of cases of trait loss. The question that we explored very briefly after that was, can you regain a complex trait once you lose it? There was a claim a few years ago that you could, and the example is in stick insects. So their argument was, if you plot flightlessness versus having wings on this tree, the lowest number of changes you can get is 7. So if I have 1 loss, 2 loss, 3 loss, and 1, 2, 3, 4 gains, that's 7 changes. That's the lowest number of changes I can get consistent with this tree. That's the best, simplest explanation. Immediately, the response, and including today from some of your colleagues in the class, was, yeah, but wait a minute. If I plot it where I only allow losses, right? Then I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, I think it was, 13. 13 losses. Is losing wings twice as likely as gaining wings? Probably more than twice as likely, actually. In that case, this makes more sense than this. It's not the sheer number of changes, it's how likely those changes are. So there's always some caution in interpreting evidence of gain and loss of traits.
And if you think about it, it makes a ton of sense that it would be easier to lose a trait, because
Sure.
there are many ways that you can lose a trait.
That's right.
And though there are, yes, many ways that you can evolve a trait, it's a lot more complex given the fact that mutations are random, that they are selected for, and all of that stuff. So highly, way more likely that you're going to lose something because of, the possibility of losing the gene, deterioration of the gene, shutting off of the gene, all of those things are much more easily accumulated than complicated series of mutations in order to evolve the trait in the first
That's right.
place.
And once you if you turn off that developmental pathway, it's no longer being maintained by natural selection to make wings, for example, then those genes and that pathway can deteriorate by mutation, and then you're not going to get all those things back to being functional by chance. It's just very unlikely. So you have to evaluate the likelihood of each direction of change. So that's as far as we went, except to give you an example of another tree to evaluate. It's one of your questions to discuss online. And again, it has to do with evaluating trait evolution on an actual phylogeny from a published paper on mussels. And so we highly encourage you to go and evaluate that online, and part of a discussion as you're studying.
So all the best on the midterm, and we look forward to getting to the second part,
Yeah.
ecology.
All right.
Take care.